What is a genome, and how large were the first genomes made up of RNA molecules? Which proteins are evolutionarily the oldest? Were the oldest genomes linear or circular? We will try to answer these and similar questions in today's episode of the series Secrets of Genes, in which I welcome you. My name is Eduard Kejnovsky, and I work as a geneticist at the Institute of Biophysics of the Czech Academy of Sciences. We often hear around us that the human genome is revealing its secrets, that the genomes of other primates have been read, and so on. But what should we imagine when we hear the word genome? The answer is simple. The genome is the complete hereditary information of a given organism, that is, all the DNA found in every cell of any organism. Experts will hopefully forgive me for minor inaccuracies when I say that every animal or plant species has characteristic genetic information, which is more or less the same in all the cells of its body. As we will see later, the genome is not made up only of genes, but also of a lot of junk or even parasitic stretches of DNA, which are found in most organisms. In all cellular forms of life, the carriers of genetic information are DNA molecules. Only in some viruses are they RNA molecules. But let's go back for a moment to ancient times, when genomes were made up only of RNA molecules. The first genomes were very small. Their small size results from the instability of longer RNA molecules. The sizes of the first genomes were also considered by one of the pioneers of scientific research into the origins of life, Manfred Eigen, who concluded that the limiting factor is the accuracy of copying the first genomes. He realized that the lower the error rate during replication, the larger the genome that can be passed on to the next generation. Precisely as a result of this limitation, namely inaccurate replication, the length of the original RNA molecules probably did not exceed a few hundred bases. However, the problem of low replication accuracy could be addressed. One way was for the genome to produce more copies of itself, thereby increasing the likelihood that at least some would be correct. Another defense was the fragmentation of the genome into pieces, along with a related evolutionary innovation, namely the combination of individual fragments through the process of recombination. It is easy to imagine that, from a large number of molecules and their mutual combinations, it is possible to assemble the correct resulting genome. It's similar to if you were manufacturing machine parts very imperfectly, and many of them were defective. But you would select only the correct ones and assemble the final machine from them. However, a considerable number of errors also had one advantage. It accelerated evolution. The first genomes made up of RNA molecules initially coded for only a single protein each. This could have been a replicase, an RNA polymerase, possibly the oldest protein, whose sole function was to copy the genome. Replicas probably arose by chance, and at first it was advantageous for the genome because, for example, it stabilized it or beneficially altered its conformation in some way. It was only later that it became capable of catalyzing the synthesis of additional copies of the genome. As a result of replicas errors, a population of similar RNA molecules arose, which became increasingly different from each other. These replicating molecules, replicants, were mutually beneficial to each other. Some coded for replicas, while others for a protein that induced better spatial organization of RNA. And eventually, a gene coding for a protective coat protein also emerged. In this way, a cooperating fragmented genome could have arisen, a kind of network of interacting genomes that were mutually beneficial. Manfred Eigen called this primitive genetic system a hypercycle. At a certain stage, the hypercycle became spatially separated from its surroundings, for example by a membrane, and a primitive metabolism took place inside. This was the moment when the first cell could have arisen and the first primitive organism was born. In honor of Manfred Eigen, the fictional organism described above was named Ryborgis eigensis. The maximum size of the RNA genome ranged in the tens of thousands of nucleotides, which is about the same as the genome size of the well-known coronavirus SARS-CoV-2 today, whose genome is approximately 30,000 nucleotides long. However, evolution was heading toward increasingly larger genomes. RNA molecules were no longer sufficient. The transition to DNA molecules occurred. A key role was undoubtedly played by the enzyme reverse transcriptase, which enables the transcription of RNA molecules into DNA molecules. The ancient process of reverse transcription continues to shape the genome to this day. Evidence of this is the abundance of various mobile genetic elements in today's genomes, known as retroelements. These spread specifically through the action of reverse transcriptase. Retroelements are first transcribed from DNA molecules into RNA molecules and then back into DNA molecules. Because the original copy of the element in the DNA is preserved, the elements are always duplicated. The new copy eventually inserts itself into a new location in the nucleus. We still know very little about the origins of the genome. It is not exactly known how large genomes were at the moment of the transition from RNA to DNA. Perhaps it was somewhere around the size of the aforementioned coronavirus genomes. It is also unknown whether the first genomes were made up of linear or circular, that is, ring-shaped, nucleic acid molecules. An interesting explanation is offered by Professor Eduard Trifonov from Israel. According to him, the first DNA genomes were still relatively small. They were composed of many small circular molecules, which gradually fused together to form increasingly larger genomes. According to Trifonov, the individual circular DNA molecules gradually recombined and fused with each other, which led to their enlargement. At a certain stage, the rings then transformed into linear molecules, and the first chromosomes were formed, containing various numbers and combinations of the original genes. The ancient structure of the genome is now difficult to uncover. However, some researchers claim that all the rises and falls of genomes were once inscribed in their DNA. It's just that time, with its relentlessly ticking mutations, has blurred them to varying degrees, so we see some events more clearly than others, but most of them have disappeared beyond the horizon anyway. So, that's all for today. In the next episode, we'll talk about the minimal requirements of a bacterial genome, how many genes a viable bacterium needs at the very least. I'm looking forward to seeing you. And if you enjoy my storytelling, I'd be grateful for a like. And if you'd like me to continue making episodes about genetics and evolution, please subscribe. Your support means a lot to me. Thank you, and bye-bye.